Our
You're
next
welcome.
speaker, we're going to have actually two speakers in the next session, Vaughn Walton and Bob Martin. They're going to be talking about the red blotch situation in Oregon. Vaughn Walton is an associate professor of entomology in the Department of Horticulture at Oregon State University. He works on a variety of important insect pests in a number of different fruit crops, red blotch. Dr. Bob Martin is a research plant pathologist with the USDA-ARS laboratory in Corvallis. Bob has been also very involved in the red blotch disease in Oregon, and they'll be telling us about the red blotch situation in Oregon. Vaughn will be up first, and then Bob.
So, can you guys see my screen?
Just need to go into presentation mode, and you're good to go.
Fantastic. Okay, so what I will do is I will I'll show you what we've been finding when we're looking at two types of viruses. We're going to be looking at leaf roll virus as well as red blotch virus, and I'll be looking at three aspects. Firstly, just kind of describing a little bit of the epidemiology, the triangle, the vectors, the host plant, and then the virus. And then I'll talk a little bit about two areas, southern Oregon and then up here in the Willamette Valley. I will also then just come up with some of the basic conclusions. So I've got about 7 minutes just to make sure that I'm sure everyone knows this, the grapevine is the host for the virus, for both of those virus types. We have the pathogen, which would be the viruses, and then we would have the vector. And so for vine leaf roll virus, we know what those vectors are, but we don't, at least not from my side, we don't have a very good idea of the viruses in red blotch. So if we look at those two vineyards or those two regions, looked at southern Oregon, as I said earlier, and then we also looked at the Willamette Valley, and we looked over at these vineyards over a period of time to see if we're seeing differences in distribution of those viruses, as well as if we're seeing spread of red blotch virus in those vineyards as well. So first, let's look at the methods that we followed. We looked at mealybugs, search for mealybugs, very, very intense survey work. We also did beet sheeting in different locations in those vineyards, so we were looking at leafhoppers, aphids, and other insects in those vineyards to see if we can find a correlation between the virus distribution and then the potential vector distribution. And then at the end of the season, we'd collect these large quantities of leaves. We would have those leaves processed, and then we'd ship them for analysis down in California by SUDI. So if we jump to the Willamette Valley vineyards that we were looking at, this is a vineyard firstly looking at the leaf roll virus. In this vineyard we found leaf roll virus towards the left side of this vineyard specifically. We did not find any spread of the virus in that vineyard over quite an extensive period. If we're looking at the insect distribution and composition, we were finding in this area no mealybugs to really speak of. We, however, did find some leafhoppers, and I'll go into some of those details in a little while. Really interesting for that vineyard is if we look at the red blotch distribution, it's a significantly different distribution. So what we're looking for here are these light-colored areas. So red leaf roll here is light-colored. Here, red blotch is also light-colored. So we can see that that distribution is significantly different. In my mind, that indicates that we have different vectors spreading those viruses in those vineyards. So the next question was, are those viruses being spread through those vineyards over time? So we did that spatial analysis, and so this is significant spatial analysis showing the spread of red blotch, and as I said earlier, no leaf roll has been spreading from the year over year, but with red blotch, we're seeing at least a doubling of vines year over year in that vineyard in the Willamette Valley, which is really concerning to us because that's one of the key grape growing areas here. If we look at Southern Oregon, year over year, this is a vineyard that was sampled in Southern Oregon, very low infestation in 2014. In 2015, we got 6 times higher red blotch in that vineyard. Just 2 observations from there. We found very, very high Western grape leafhopper numbers, as indicated by these white numbers here. The other thing, similar to what Keith Perry was showing, we were also looking at surrounding vegetation. We found 1-year-old seedlings, which also had the virus within itself. We didn't go into the details as was done by Keith and some of the other people, but that to us indicates that it's most likely a very highly mobile vector that's spreading it. So if we're looking at the epidemiological trends in those vineyards, when we look at the Willamette Valley, this is not all the data that I'm showing here, but we're seeing at least a doubling in the vines, which has red blotch virus there in the Willamette Valley. And then if we look at Southern Oregon, we have at least 6 times more viruses. If we look at the Southern Oregon vineyard, we were finding very, very high numbers of leafhoppers in that vineyard, as I was saying earlier. If we're looking at the distribution or the species composition of those leafhoppers and all the insects that we're finding there, we're finding that the majority of insects there would be Western grape leafhopper. Of note is we had several plant hoppers and tree hoppers and so on there as well as aphids, and I don't have the time to go into those details, but we're getting some really encouraging results regarding the distribution of the virus and then correlating that with some of these insects that we're finding here. And again, I don't have time to go into those details. So that's basically a summary of what we've been finding in Oregon at this stage. We've been getting funding from the Oregon Wine Board. We have some funds from the Oregon Wine Research Institute, and then of course our collaborations are very, very important collaborations with our California colleagues and some of our USDA colleagues as well. So that's basically a short summary of what we've been finding. Not enough time to share everything, but that's basically it. Thank you.